We'll start speaking about Arkitekt today, which is sort of my baby, as Lena said, that I developed during my PhD and that I just waned off last year, and that I'm excited to present today. So thanks a lot for the invitation to let me talk about it. Yeah, I will just jump right in. First, I will tell you shortly about me. As Lena said, like I'm a clinician scientist at the Johannes University, and there I work in the Department of Anatomy and Cell Biology. That's the sort of research part. And then I work mainly now in the Department of Pathology, where I work on the clinical sites. But sort of these two paths are now sort of merging because a lot of the work that I'm doing is on bioimage analysis. And that's also what I did for my work when I was still at University of Bordeaux, where sort of this software platform that I will introduce to you originated. But yeah, now at Johannes Kepler. At that, like in my new position, which is a bit different to my old position, I'm more working with biomedical personnel, mainly with life scientists. So in Bordeaux, I got the chance to really dive into bioimage analysis, and now I have to apply it and bring it to the people, I would say. And that mainly means that we are working like with preclinical data. Wait, I will just use the pointer. I don't know if you see that. Is that? No, that's the pen. Sorry. That we work with like preclinical data, mainly neuronal, like the Department of Anatomy is mainly invested in neuroscience. So we work with the axon initial segment, but that's not the topic of today. And then on my clinical side, I now work more and more with like histopathological slides. So that's like a cytology image. But what unites these both is that I work with the personnel to streamline their analysis and get some biological insights or medical insights from it. And then what I feel like the most important part nowadays is that I focused on this last part of actually looping back what we find biomedically to improve what we do acquire in the first place. So that's what I do on a day-to-day basis. And how does that look like for most of the work is that for most of the life scientists in general, so that's like based on some statistics from Sigvaranothan in 2013, they surveyed a lot of life scientists and it's the same for us. What do people use? They use the Microscope software for their analysis mainly, and then they use Fiji for the sort of workhorse for anything my biomedical image analysis, and then they export their data for statistical analysis. And as most users, they do this completely interactively with a lot of manual labeling steps in between. And that's really great for the most part of the university, for the university departments. But I cannot help to think back a little bit about, sorry, no, this doesn't work. Yeah, sorry. But I can't help to think a little bit back at my time in Bordeaux, where we didn't use a lot of those classic Fiji tools, but we developed our own analysis pipelines and used a little bit different tools, I would say. So what we focused mainly in Bordeaux was automated microscopy and bioimage analysis. So we there worked with like really high throughput analysis where we had microscopy samples that are mounted on a multi-well that we monitored with high resolution microscopy and then used advanced bioimage analysis tools like CARE. I don't know if that's familiar to some people. By the way, if you have questions, please scream away. I would love this to be as interactive as possible. There's ample time to interrupt me. I just did a presentation, so if you have questions, just go ahead. I will try to look at the chat and the images. Cool. If you have somebody also, like, just really scream out. Um, exactly. So they use tools like CARE in our old team, my old team, and Stardust to really segment data and automate that whole data process. And what I liked about it is not only does it sort of alleviate the stress of labeling data yourself and all of the manual labor, but it also sort of reduces the human bias in our analysis. And also there, we bridged a little bit things further and went into this sort of smart microscopy realm. I'm not sure if people are familiar with the term, but sort of what smart microscopy is brings sort of the analysis very close to the microscope. And instead of like just going image, then analysis, and then at some point results. It sort of integrates the whole analysis process. So that, for example, as illustrated by Mahejish, you could, for example, imagine a workflow where you start monitoring specific populations under the microscope. And then once you detect an event here, a mitochondrial fission event, through automated analysis, you would start really acquiring the image faster to capture that event. And so through this process, you can really sort of defy the holy grail, the holy triangle of image analysis, where you always have these trade-offs. And yeah, so why are people not really using these new workflow paradigms? Because there's an ample of tools that are helping us now to do this, and they are part of a new spectrum of bioimage analysis tools that live a little bit outside of the ImageJ ecosystem. So some people might be familiar with Napari or Stardust, like Python-based applications, or tools like Micromanager that still interface with ImageJ, but use sort of like allow you to steer the microscopes better. And these tools are really great, and the developers are really great, and I'm so happy that I could integrate a lot of them in my thesis, because they really allow you to interface with the hardware more closely and to tailor the analytical process more to your biological question. Instead of like standard ImageJ workflow where you have to go through specific steps or macros, they are really allowing it to be more tailored to the questions. And generally, life scientists, like the people in Kepler University, they love these tools. They also know about these tools, and that goes like in all of the surveys that is shown. But what we also see is that there's still a massive gap in the sort of non-expert space and the expert space. So there's a self-declared non-expert bioimage analysis experts. And you see that like tools like Fidget dominate that space, even though like tools like Cell Profiler and Napari are getting a handle. I don't know if anybody knows Napari. I guess some people might have heard about it, use it. But some of these expert tools still remain very much in the expert space and are rarely used outside of collaboration efforts. And I was wondering doing my PhD, and I'm still wondering why that is. Why don't people use these tools more often? And I would like to illustrate what I feel could be reasons for this on a few slides. And that I would like to do with the help of Kim, which is a standard life scientist, like a model life scientist, a fictive life scientist that could go at any university. And Kim is interested in sort of the tribolium model organism that she would like to analyze. And she's especially interested because of a fictive reason to analyze the cells at the hind of the organism and at the front, and has some interesting insight that there might be a biological interesting fact about these cell sizes. And so she devises a Fiji script that she uses for her analysis, where she just marks the cells, does a max protection, then some thresholding, and then she uses the Fiji analyze particle plugins that a lot of bioimage scientists use to sort of know, okay, what's the size of that particle? And now Kim is really excited and very happy with the workflow. It runs super well. But she is also realizing that there's some phototoxic effects that she does through her imaging on the cells. So she would really like to decrease these effects, and tries to make the images a bit less, with less laser power and less pixel dwell time, which ends up that her images all of a sudden look like this, and her whole analysis pipeline sort of falls completely apart, because all of a sudden the max projection gives us the pixel values and the thresholding and the analyzed particles just don't work. So Kim is a bit disillusioned, but luckily she attended one of those Maya GT webinars, and she heard about a few technologies that are like getting into the market that could really help her. One is Napari, that she knows would really help her visualize her 3D stack more better, and maybe help her identify the regions that she wants to really mark. And then she heard about this CARE, content-aware image restoration, which is a denoising algorithm that was published a few years ago and showed great promise with exactly the same model organism that she tries to use. So she sets out to develop like her own workflow that she would really like to do. And that now would include like sort of marking her voice in a party using the 3D abilities, using care as the denoising part, and then sending the stuff back to Fiji as she used to do the maximum intensity to protection and this sort of Otsu-based thresholding, and then use the analyze particle plugin, and then look at the stuff back at Napari. So she's really excited about trying these tools out, but little does she know that she will meet the what I will call the apocalyptical horsemen of modern bioimage workflows, which are like portability, interoperability, orchestration, and especially data management, our last big horse. Because when she tries to install all this software, she soon realizes that these tools have very specific hardware requirements, which come to the point because they were developed to take advantage of these things. So, for example, Care does not run on her old laptop that she uses for analysis because it needs a modern GPU. And therefore, she needs to install it on another lab computer. And then Napari and Fiji work together well, which is nice, but some other tools that we know can't even be installed in the same Python environment, and it's really hard to install them on the same machine. So what she ends up is trying to install these things and doing it on multiple computers in her lab. And then comes this other horseman quite soon when she tries to run her first workflow because she installs these apps. And that's that there's this a lack of a universal data standard in modern bioimage. So her image comes originally from a Leica microscope, and she can open that in Fiji, but her Napari version doesn't allow her to install the bioformats reader, so she can't really open it in Napari. And then for Care, she needs it in a specific format. So she writes her own Python script to do that. And then for Care to put it back into ImageJ, it's the same thing. So she tries to make this whole pipeline work, but she has to write a lot of little helper scripts to make this pipeline work. And that is a problem, like it's a very common problem because like no universal data standards exist. And even the open file formats like OMTIF and OMNGF, OMTSAR, if you haven't heard about them, they're super good and they're going to change a little bit, these things. But even they struggle with cross-tool compatibility because some tools interpret the standard differently. And what I especially care about is that even if you manage to convert the files, often the metadata and the annotations on that data are sort of lost. So when you end up with your cell CSV file, you don't really know anymore where that image file came from. And it makes these tools really interoperable. And not only that, is that her once really interactive one-click in Fiji pipeline is no longer interactive at all. Like she needs to now run these different applications and spend minutes in conversion, running from one computer to another, converting these files again, running it. And it really hinders her in the timely orchestration of her workflow, and importantly, to the time to feedback if something went wrong in her analysis pipeline. She doesn't really have a way of managing that. But she managed everything, and she's happy with her pipeline. She gets a result, and now the paper's published. And then a few months later, she gets contacted by some people who would really like to use some of the data. And that's where she meets the last horse of bioimage analysis. That's the data management problem or the lack thereof. And that's because now that she has moved her files from 2 different computers, 4 different applications, her folder structure may have made sense in the past, but 5 months later doesn't really make any sense to her anymore. And she's not alone because data not shared is the number one, data not found is the number one reason why bioimage analysts are not sharing their data, which only happens in 30% of the cases. So there's been a lot of initiative in the past to establish a way of doing this, and these are sometimes referred to as the FAIR principles for findable, accessible, interoperable, and reproducible data. But tools that bring this in, like bring these features in, are rarely employed. Some people might know Omero as a database, which sort of centralizes your data storage and brings it into like a format that you can use and explore in the future. But this is often like a post hoc solution, and it's not part of the workflow itself. So the data management is always like an archive solution instead of a real world data. So Kim is a bit disillusioned now with these modern tools and would just love to have come up with the idea of using the tools, and would have had some person in the lab, preferably not the bioimage analysis guy, to help her out with orchestrating these tools and actually running these different applications for her, and have sort of a middleman that she just upload her idea and that helps her orchestrate. And she's in luck because we do have actually a lot of solutions besides the solution that I'm going to introduce. And there's tools like Bioimage IT, which I think I saw Arthur Masson in the things. She's part of the team that are really great tools to start orchestrating multiple bioimage apps, self-contained, and run them on a local machine. Or tools like EMJoy, which is quite popular, that allows to really orchestrate also different workflows and connect to your microscope. And then there's sort of general workflow managers that also exist that run on clusters that help you really automate larger pipelines. But they sort of lack in some regards, and I'm happy to talk about this in the discussion, why I feel like I felt like there was a need, even though they're great tools in general. So what I'm thinking, like, there needs to be a holistic approach to managing this. And this is where I sort of developed Arkitekt, which is now the subject of this, was subject of my thesis, subject of the paper. And with Arkitekt, I tried to tackle this problem. And the way I did it is a bit different, that I feel like Arkitekt was not developed as a tool. So the core idea of Arkitekt, that it's a software platform that connects to the tools. Like, instead of encompassing one tool that now would do all 5 tasks at the same time, it sort of tries to connect and manage modern bioimage apps in a universal workflow. And it does this by sort of centralizing it as a central server app that you install in your lab, or hopefully in the future, if everything goes right, you can just connect to it in the cloud. And then your bioimage apps that you have in your lab can connect to this central repository and advertise functionality that it has. So for example, your microscope would say like, I can acquire 2D images. The star says like, I'm able to give an image. If you give me an image, I am able to segment the cells and give you a segmentation map. And Ampari and Fiji have their own functionalities. And sort of Arkitekt gathers this sort of ideas, or like sort of nodes, as we call them, or functionality that all of your apps in your team have. And then it allows you to orchestrate these nodes into workflows. And I'll show you how that happens. So you can then wire them together and say like, hey, I would like to acquire the image. Then I would like to segment it. And you just wire nodes together in a sort of graph. What also sets Arkitekt very much aside is that it implements a sort of data storage, a central data storage. So if these apps are acquiring images, they don't store it on a folder on a computer. They actually store it in the platform itself in a database. And sort of you have a way then to explore your data through the same web interface that you use for accessing this server. And you can like interact with your data then. Does that make sense for everybody until now? I guess I see only, yeah, good.
Yeah, yes. And
One thumbs up, that's nice.
in the
So,
if there's some question in the chat, I will ask you if people don't want to ask you directly with the microphone.
okay, perfect. That's enough.
But we have a question from Clément.
Okay.
How do you deal with container environment problems? For instance, if you call several Python program requiring different version of same library in the same workflow.
Yep, perfect. I will get to this in a second. That's the first question that we'll answer after the mini demo, like the mini video, if that's fine. Okay, so sorry, I will just go back. So before I bore you, I had to have this presentation a million times with life scientists, and content environments are great. Good question. But I like just to tell you what it actually looks like. I've recorded a little video with like an older version of Arkitekt, but it captures the idea. So let's see what Kim's workflow looks like. So when she does this, she can go, like if she wants to create a workflow and come up with the idea, she just goes to the Arkitekt webpage and says like she wants to create a workflow that that orchestrates her task. And then because Arkitekt has these apps that are connected to it, and these are now an app like a stream ROI, which would get the ROI's marking process in Apari, and just whenever you mark a ROI, it will send it down the pipeline, so to say. And then she can say like, drop that image, denoise it with care now, do a maximum intensity projection. And then she would like to use in Fiji or any other program, do the thresholding of that image, and then run, for example, the particle analyzer from Fiji. So she searches for that functionality, and now she has all of these nodes in the workflow pane. And now to sort of orchestrate this task, she just has to connect them, do like creating this data pipeline. And let's see just how this happens. Like you say, the image that goes out will now be denoised, then it will be maximum intensity protected, then it needs to be also thresholded, and then use like the analyze particle plugin. As we all know, bioimage workflows are not that simple. Like there's a lot of parameters that you often have to fine tune, but you can set parameters like, for example, the plugin parameters already in this online workflow. So once you save them, they will be applied to every image that you run in this pipeline, for example. And you have a sort of predictive way of saying like, okay, what should happen when in my workflow? So that's a way of constructing it. And this process has also been completely streamlined in a newer version of Arkitekt, but I'm presenting you the old one. And now she can actually, run this workflow. So now what we see here is that she has Napari open on her computer, Napari, this 3D visualization thing, and Fiji. And these two apps are connected to Arkitekt. You see this lower window down there. It means like the app is actively connecting. And now she can run this tribolium analysis workflow on the app. And she just says like, okay, which deep learning model for care does you want to use? And then Arkitekt starts by sending the image directly to Napari. So we see that open now. On top of that, you have this sort of progress window in Arkitekt that allows you to see what is happening in your workflow in real time. It's especially important because some of these apps, as we will see, they run internally on the server and they don't run on your computer, and you can inspect sort of the results in between. So now Kim marked a ROI, and you see once she marks the ROI, it gets analyzed in Fiji. Let's see it again. Like she marks the ROI, it goes through this pipeline and goes predict care, maximum intensity protection, Otsu, and now the results are displayed in real time at her fingertip in Apari. Fiji is right now open, but it doesn't need to be open. Like it could be running on another computer. It doesn't need to be run on the same computer, and she could do everything, so to say, in Napari itself.
I have a question about this step here. When I design a workflow, I create the workflow step by step, but before having the whole workflow, I need to check if the settings are good or not. Could we make some stop in the execution of the whole workflow saying, here, give me a stop, let me show the result, let me fine tune, restart, 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 without cutting the workflow in many parts, in many different little ones?
So you mean like an interrupt, so to say, to
Yes,
inspect
like a
results?
wait for user in your big Fiji macro, you make a wait for user, you check, you manipulate, and when you find, you continue or you cancel and you restart.
Exactly. So that's perfectly fine. Like, for example, there's a way, because all of these apps can be completely interactive. You could write a node that says like load it into image J, and then the user does some sort of manipulation, and then you send it further, like adjust contrast, for example. It goes a little bit against the principle of what I would like to establish with this, because now the data sort of is managed by Arkitekt, right? And I know that's maybe I will go and discuss this later, but I know what sort of tasks have been done. So if you do an all catch all task and you adjust anything, I don't know what you did, so to say, like what operations you did on your data. And that goes into this sort of data provenance aspect that is integrated in the platform. But it is definitely possible, and I use it exactly like this for the most part, because a lot of times you need to fine-tune examples.
Yeah, and this is exactly the same question. I think, if I understand correctly, exactly the same question in the chat. We need to first fine tune it. If you have an already working pipeline, you know it will work. So you do it and you apply it. but we need first to construct it and check it before applying it.
Yeah. So you can also run every node by itself. But the magic, I really feel, this is why I put a lot of effort on making these apps standalone. Like it's Fiji as you know it, right? So if you load something into Fiji, you can do whatever you want. At the end, you just need to say, send it back. So that is definitely possible. Yeah.
Yeah,
So
I
yeah.
also have a question. So you need to install everything. You didn't show the installation part of the architect. So
Mhm.
do you need a Docker or do you need do you
Yeah.
need to have everything on your computer or how does it
So
work?
this is right now how it works. So right now, Arkitekt is sort of a Docker image. So like you need Docker, which is a sort of virtualization engine to run all of the Arkitekt services and to manage dependencies. So that's sort of a thing that I use internally a lot. And if you want to install Arkitekt on a server, you need this Docker environment, which is pretty easy to install nowadays. But there is this sort of step that you need that definitely on one computer in your team. And then for the rest of the apps like Fiji and Apari, you need to install plugins for both systems in order to connect to Arkitekt. So there's an initial cost of installation that we would like to get rid of with the rest of the Arkitekt developer teams. So this thing that we have a central Arkitekt instance that people can just connect to, but that's not there yet. So for now, this is the sort of step that you need to take.
There's a
And
question in the chat too that's called it's just an IV question. What
Yeah.
is the difference between Architect and IC? The IC software. I don't know if you know that.
Yep, I know IC. So IC is pretty much also allowing you to have ImageJ and have little blocks itself and run these analysis tasks, which is super cool. And it allows you also to have Conda environments. What is a bit different is that IC is sort of running everything on your, as far as I know, like running everything on your computer. And it is Java based, so it's sometimes a bit hard to add plugins, but there's this sort of idea that it runs all on your computer, which is a big benefit. But for example, if you want to connect your microscope or like a GPU server that sits somewhere else, you're not really able to do this. So Arkitekt sort of offloads tasks to different hardware in your lab or somewhere else. So that's a bit the main difference that I would say.
and
But IC is a great tool. Like it also facilitates, it has ways of sharing workflows, and I adopted a lot of their ideas into Arkitekt. And I think for now, it's the better solution if you want to run things locally. I'm hoping that Arkitekt will get to the point where IC is. Let's put it this way.
Another question is when you install Arkitekt, so I think it's operating system agnostic, so you can have Linux, Windows, or so for the server too. We need just to have a virtual machine environment, for example Linux, and I can put all of my Architect on it. And I need to install my Napari, and I need to install my Fuji and connect it. And for
Yep.
example, as Clément asked about Conda environment, sometimes Napari, it's more for simple plugin installation, because if you want to put a multi plugin, you will have problems. So in the idea of
Perfect. Next
Arkitekt,
slide.
cool. You can put many Napari environment and say to Arkitekt, do this. And another question is, when you show us the film, you type Outsu or thresholding and give you how does it list that? Do you have to put it in Arkitekt, or does Arkitekt recognize everything into the software, list it, and then give it to you?
Yeah, very good points. So this it's going a bit into the technical realm, which I'm very happy with. So just for this, what I want, for example, like Napari now has a lot of plugins that allows you to read image data, maybe run even a deep learning algorithm itself. But the problem what I see with Napari is that like once you have Napari, you have to have very specific requirements for each and every plugin, and then Napari itself at some point can't handle to manage all of this. So what Arkitekt tries to say is, hey, Napari, you're good at visualizing stuff. you don't need to run these plugins yourself all of the time. Like some of them are great if you visualize it, but what you can do is you can offload, for example, the deep learning task into an Arkitekt plugin. And plugins are now, again, completely Dockerized applications. Like they have all of the dependencies inside, and they can be installed through a simple install from the App Store. So there's an Arkitekt app stores where these plugins are. And what happens when you start them, they download it onto the server or like an engine that can run it and runs then the plugin there. And so Napari then in the workflow, so to say, would call this plugin and get the result back.
Okay, so could I just sum up if I understand correctly? Instead of doing Napari Viewer, install all plugin, you go into Arkitekt, install the plugin, and it will have the list of plugin in a Docker, so it have different environment for each plugin. And then the Napari Viewer just connect using Arkitekt to the plugins you install.
Exactly.
Okay,
so it
cool.
installs it, runs it, and then it sends it back for
very smart solution to have a Napari with many tools at the same time.
So the same is if you have Napari, for example, and you want to run a Fiji plugin, you need that Fiji plugin, you call that Fiji plugin, and you also send it back. Like it doesn't need to be anything. Like you can use Arkitekt, the workflow interface, but you can start a workflow, and this is then a mini workflow, which just says like, use the currently run image, send it to Fiji and go back, and you can start this from any application. And then you have that, for example.
And another comment from Clément says, so Arkitekt App Store contain all Napari and Fiji plugins in the end.
Yeah, so that's a very good point. Arkitekt, if you want to create an Arkitekt plugin, what you need is to create this node. And this node is sort of just telling Arkitekt what it expects to happen, right? It needs to tell, hey, auto thresholding, my input is an image. I maybe need like a parameter, like the hysteresis parameter or another parameter, and I give you as an output another image. So this is some information that I need to tell the Arkitekt server from my app. So what happens is that every app has sort of a SDK, like a software part, that actually tells its functionality to Arkitekt. Does that make sense a bit? So in a sense, the app itself tells, hey, Arkitekt, I can do this task. And then it is an Arkitekt, and Arkitekt knows then, well, now if I want to run this task, I need to tell this app to run that specific code. But that needs to happen. Like I can't just have all of the ImageJ plugins automatically part of Arkitekt, because I need to understand what is their input and what is their output. You could run what you mentioned before, this step where you load an image in, you run whatever you want, and then you have it out. That is possible. And you say like, my input is an image and my output is an image, and in between I do what I want. But what I really would like is to have more fine-grained control. Say like, I run this plugin. And right now I had to write major parts of this to just like wrap it with one line of code to say what the plugin actually does. And that needs to happen. But there's like efforts to run, for example, Nextflow or Galaxy plugins and have that definition as part of the Arkitekt definition. But right now it needs to be happening by hand, pretty much, or programmed.
And I have the same question as Clément. It's a user, the setup to add the Otsu, the different plugin you want to use is something that you have to be done by the users? Or do you manage the fact that you want this specific tool of Fiji for your Arkitekt?
This I didn't like, again, sorry, like this I didn't understand. Does the user have to?
The Arkitekt, the list of the things you call in the film you put, you ask Otsu, you have Otsu, you ask analyze particle, you have analyze particle. But if Arkitekt doesn't take it automatically into Fiji, it just asks Fiji to do this and take the results from Fiji. How do you list it into Arkitekt? Is it the user that have to put it?
No. So the app itself, like the sort of plugin, the Napari plugin or the Fiji plugin, has coded in what functionalities it has. So instead of Arkitekt knowing all of operations that exist, every app and the apps that you develop itself can tell Arkitekt, hey, I can do this. So I can say like, I can do auto thresholding. And then Arkitekt just knows, well, there's some app in this in your whole lab that can do this one task. So you could develop, and the developer interface is quite straightforward in Python. You could develop just a Python function. It is literally that, and says like, I take an image as an input and I have an image as an output, and then you register this. And this is something that you can run now in your workflow. That makes sense.
Okay, thank you.
So what is also important that, and that is different from Arkitekt to some other types, is that the data is stored centrally. So every app, like if you have, for example, an image and you send it from Fiji to Nepari, now there needs to be a contract, so to say, between the two apps, what an image looks like. So Arkitekt enforces very strictly what the image data type is. You can always construct a more broader one, like an array one or whatever, but it enforces it on the API level. So you can't wire together a Fiji app that takes a completely different image type. You need to convert it on the way before, if that makes sense. Exactly. What is also important in this workflow, and that's something that is a bit different to also some of the workflow managers, is that all tasks can run independently. And that will become a little bit important in one of the tasks that I will describe later. So that, for example, if you have something that takes a long, long time to do, and some fast tasks, they run completely independently. You don't need to wait for, imagine you have a task where you want to just segment the cell bodies and then run some analysis task, but you would really like to have some feedback before. The tasks that take longer can take longer, and you can process your data already. And they run in real time. So there's no delay, no scheduling. Once you mark, for example, this ROI, it gets sent through the whole pipeline immediately. And the next one, the same. And yeah, everything is stored centrally in the database and interlinked. So for example, if you have a ROI that you mark in the image, I know that you marked the ROI in that image, and I use the information from the Nepari where you marked that ROI, and it's like in the image database linked to another with its metadata. And you have like multiple users can comment on stuff. They can, in the new version, interlink interlink data points in even a graph database. That's a bit more technical, but it allows you to really construct large like knowledge into your data. And what I feel, and this is a little bit what we just discussed with this running a Fiji plugin that does everything. It is possible, but what Arkitekt also does is like it knows what you did to your image because it knows what the node will do to your image. So it keeps a record and says, hey, this ROI that I have marked here, I cropped it from the original image. I used the denoiser in care, and it knows exactly which care version you use at what time with which model. It knows which Fiji version you used, and it construct this sort of provenance, data provenance pipeline, so you know what happened to your data. And if you want to go back, you know this ROI, you can go all the way back to see like which image it was and what did I use. This is especially like afterwards nice if you just, in your method section, you could put this like provenance thing and it would give you people idea what happened to your data.
And you can you have such a sort of I don't know, PDF or Excel or text output that give you this information
Yeah, so
just
it's called a run.
for the publish it in the
Yeah,
end.
so it's called a run. Actually, a run is sort of a JSON representation of all of the tasks at which point. And like in the old, like right now we're doing a massive overhaul, so I cannot show you this feature now, but you can export this and put it in a website and people can just go through the time, what happened, using the same interface, sorry, that we saw here. So they have the same visual representation. They can just go through it and see what happened with each data point. So that's something that you can publish. The problem is you need to also give access to your data then, because this run is just like a sort of a reference to these different data points, but it doesn't show it.
Okay, so you just have to take few data for the publisher to have to give some try and have some even for
Exactly.
reproducibility, you will not put all the data you use. You can put a set of data saying I have this result or set of data, try to reproduce it.
Yep, exactly.
Okay.
The workflows themselves, like there's runs, which is like what you did, and workflows is also like the recipe. They can also be shared. Like you can export them and load it into another computer. And the nice part is that they're a bit agnostic. So you don't have to have the same ImageJ version as the other person, right? you have something that can do thresholding. It doesn't even need to be ImageJ. You could have a plugin that runs also thresholding somewhere else, and you would be able to run it. So the idea is that these sort of nodes are a little bit more agnostic of their implementation. If you want to, for example, have a show node, you can run this on Fiji or on Apari. It doesn't matter where you look at it. It's still the same image. Yeah. So in a sense, I didn't go into all of the details now, but otherwise I will not get to show you a little bit of these things, that the apps can, Arkitekt can interact with these apps on the hardware that they were designed for, and they can span now multiple tools. And Arkitekt takes a bit care of this interoperability aspect of using a common file format. And because we're doing this all in real time, you can have the interactive workflow, right? You can mark it in Napari and look at what you marked immediately instead of waiting until you see the denoise results a few weeks later once you get access to that computer. And it manages the data no longer in folders, but on a central database that you can explore and share with your team if you want to. Exactly. But as I said, I like this. And this is one of the core motivations what I try to do is really I wanted a tool that makes sort of interactive workflows better. But there is more. And we actually established, and that's something that we go into the paper, but if you have rather questions, I can also skip this part, but it sort of illustrates a bit of the abilities of Arkitekt. So that you, for example, we have this high content screening approach where we have these large populations, biological samples, and we want to monitor them in an automated pipeline. But if we do this even batched, because we have very large images, like a gigabyte size, and one experiment is like 2 terabytes of data, it takes days. And we are just starting to get a lot of nice results, but we have to have a lot of time until we actually can feedback on our experiment that we did 6 days ago. Like imagine you had just one chemical wrong and you don't know what you did. It's taking a long time. So with Arkitekt, we try to do this and have now, like, it's still running in the lab in Bordeaux. Like we have a central instance that runs the Arkitekt server, and we connect our microscope, and that's commercial microscope software. That's Metamorph. I don't know if anybody's still using it, the horror show. But it sort of just uses the images that Metamorph saves and offloads them directly to Arkitekt, which then converts them, measures some meteorology data like the cell, not the cell count, sorry, the intensity, and then sort of scales it down because these images are so big that we have that they don't fit into a GPU that we need to run Stardust on. And then we send them to one of 3 computers that does the actual segmentation. Because in our analysis, the biggest bottleneck like is the Stardust segmentation, which takes 2 minutes. So if we produce an image every minute, at some point we can't keep up with our analysis. So what we did with Arkitekt is sort of use 3 computers, and it uses like Arkitekt just sends one to one, the next one to this, like a round robin, and then sort of has the analysis, and then it runs the rest, does the segmentation, and shows it to the user. And this is done, again, through this visual workflow pipe. It's a bit more complex. It's the same idea. You have an app on the computer. You have this OME microscopy node that converts the Metamorph format into like this open standard. And then you run a max intensity pipeline where you just say like, okay, what's the maximum intensity? And you do this cell analysis part. And the nice part is this runs like in live. And because Arkitekt is a web platform, you don't need to sit next to your microscope to look at the results. You can just like have a dashboard that displays you in real time what is happening. Like what's the what sort of thing are we looking at? What's the cell count? And it sort of populates this. Advanced, this is sped up, of course, because it's not that fast, but I don't want you to wait for a minute for the segmentation. But sort of displays then the cell count with stuff. And you can look at this from home, like from anywhere where you can connect to your server. And yeah, so now we can have this sort of delay for 1 week to really live analysis setting. Like we get the data now and we know if something goes wrong or we go out of focus, we can stop it. And it really helps us manage this big data loads. and especially our biologists to iterate faster on their setting. But you still have to do it manually. Like you still have to cancel the microscope itself, because it's not like you can't interface with Metamorph. Like we have a few tricks that we can, but it's not nice.
This is a question I would ask you about. You call about micromanager. Will Arkitekt manage to play with micromanager in order to control the microscope in the pipeline with the output results? Smart microscopy. Cool.
Perfect. Exactly. So that's what we actually do. And this is something that I'm also now adapting into this pathology setting, where we try to do it on cytology. So this sort of smart microscopy paradigm is often that you have a large population, like a cell culture on a petri dish, and you're really just interested in, say, for example, in our case, we were interested in the clusters of these cells, how they form over time, if they deteriorate. But we have a very large field of view where we want to monitor these clusters, but we're actually just interested in a very few cases. So we either were faced with the idea of acquiring everything, but that means that we have to acquire 2 terabytes of data in the resolution that we want, and almost 30 minutes per time point, which was really not the way to monitor these clusters. So what we try to do is establish Arkitekt as a pipeline, and that is actually using Micromanager. So what is happening now, and it's again in this paradigm, is that the microscope with Micromanager as a thing advertises to Arkitekt that it acquires 2D images. So it is able to acquire a 2D image, and it's able to acquire also a 3D image, and you can check which objective you want and where to move and all of these things. So like it has a bunch of functionality that we provide, but one is just like go to this position, acquire an image. And then the microscope does this, and because it's a sensitive setup, we don't run any of the deep learning to detect these clusters on the microscope, but we actually offload this to the Arkitekt server, where we have like again the Stardust module that we used previously for the detection of our 3D cells, and we use the same algorithm. But now we also have sort of a mark clusters algorithm, which is just a DB scan algorithm to detect these clusters. And once we have them, we go back to the microscope and say, now acquire, now with the center point in here, acquire the whole thing in 3D. So that's the sort of idea that we have. So Arkitekt is taking care of the orchestration, and the micromanager so we use Picromanager actually for this, because it was just easier to program. It does sort of the tasks. So you set up the whole acquisition in Micromanager, like interactively, how you used to, and then you just start running this script. And Arkitekt uses the information that it has in the metadata, which is important because we need to know which objective we use to do this translation back from image space to microscope space. And yeah, the whole idea is, again, this thing is just a drag and drop workflow. So once these nodes exist, you can just wire them together in a workflow. There is this caveat that you need to write this node, right? You need to know what the microscope can do, and you always have to have this plugin first. And they need to be sort of translatable to other workflows. And that is really something that is not to be underestimated, that sometimes this visual workflow planning, even though I love it, sometimes fails short. But you can always use the Python API to do the same thing if you know better what to do with your images. But yeah, so you can do this absolutely no code smart microscopy with the platform. And yeah, I'm really like, if there's anything that I feel like I would like to survive from Arkitekt, even though Arkitekt is surviving, is this approach that the microscope does one stuff good, let it stay with that task. Don't try to put deep learning in the microscope software or into Napari. I feel there's a place for all of these things. Exactly. But what we did now, like with this approach where we only scan like in a very low resolution, the whole thing, and then we detect the clusters, we're able to get the whole thing down to 24 gigabytes instead of 2 terabytes and only 7.2 minutes for the whole time point. And you can devise a strategy how you want to scan this. Like if you're really interested in the real-time events, then you can also focus on it longer. So it allows this whole thing. And importantly, because we have this central storage with all of the metadata, and we track the clusters in real time, we can now just look at the cluster over time without doing any image registration or anything else, because we're always at the center of the cluster. Good. So to conclude, because I'm not sure how the times, and I want to leave enough time for, for questions. Give you just a brief interlude for the too fast. So now Arkitekt is ready to deploy. There are versions right now that you can install and work with, even though I would advise you to wait a little bit because there's a new version coming, which should make it a lot easier to install and a lot more user-friendly. But there's sort of, it exists, and you can do interactive analysis workflows like we did with Kim's workflow. And it allows for like the streaming analysis where you really have large data sets, like we're talking really gigabytes and terabytes, and it can handle this quite easily. And then it has this sort of no-code smart microscopy. And before I let you out, I would have stopped with this in the past, but now we are having more and more people actually starting to use it and sort of using me to sort or helping me to sort out a lot of bugs that are still in the software. So I'm not saying that Arkitekt is the pristine project like ImageJ and it's that user-friendly yet, but we're working on it. And there's now more and more people that are sort of adopting it, including to run it on larger computational structures. Where people, like, you don't need to run your own Arkitekt server. So my goal is to say, have a few deep learning algorithms that everybody can access and send the data to the server, run this deep learning model on it and send it back. So that's sort of what we're working on. And we are heavily invested with OpenUC2. I don't know if you know that one. So that's a open source microscope, not a microscope control software, but a really completely open source microscope that uses like 3D printed parts. And they are trying to make Arkitekt bit their control software. Because you can imagine you have these different aspects and then you just write a workflow where you actually offload your task and really make it in a smart microscopy workflow. So if you're interested in helping out or just giving feedback, and if it's just saying it's shit, I didn't get it to run, please just drop it in the GitHub issues. Because especially the installation right now is the biggest hurdle. I understand that it's still a problem, but that's the major focus on the next platform to make it really usable. Because even though I tried to make an intuitive design, I think I failed a little bit. But we're working on it. It should be better nowadays. But I'm like, I can only tease the new version, which will come out in the next few months. And then I would guess it's nice to give it another try if you were a bit disappointed by now. But we're working. So without further ado, just a few acknowledgements of the different teams. So I was actually mainly part of the Sibarita team, even though on paper, Valentin was my PhD supervisor. I mainly worked with JB Sibarita and the whole team, which were a lovely time in Bordeaux. I loved it. And now I'm in the new team, in the Engelhardt team, which you see down here at the JKU, that we collaborated a lot. Like, that is now my old home and my new home, and we're really bringing this now to the clinical setting here. And then I would really like to especially highlight these three people down there that approached me at some conference when they heard about Arkitekt and now are developing for it, which is really cool because without these people, I wouldn't survive. Okay, thank you so much. So I'm happy to answer all of the questions.